First, this is a 1-year anniversary for me of being brought into the autism community. Thanks in part to Pat Levitt's suggestion that I be included at the Institute of Medicine meeting a year ago today on environment and autism. And in that time, I've really undergone a tremendous immersion course in autism, and I have gone to more meetings on autism than in all the other fields that I work on combined, and meetings of all sorts. And one of the messages that I think is important to deliver, and that I deliver in other contexts also, is that this is a very bizarre community. That in the research areas in which I work, otherwise spinal cord injury, cancer, a whole number of areas, there's much competition and there's much antagonism. But I have never seen such a circular firing squad as exists in the autism community. And it is important to say, Jenny McCarthy is not an enemy. The Defeat Autism Now Coalition is not an enemy. These are people who are potential friends, who are potential colleagues. They are often parents who are fighting for their kids, and they're pursuing paths that we, the mainstream medical community, have failed to offer. And they are providing information that I have to say, with my own interests in physiological aspects of disease processes, have become of striking importance. These are the enemies. The enemies are ignorance and arrogance. The enemies are how little we know, and our arrogance often as scientists at thinking that we know best how to figure things out. But we know virtually nothing at the critical junctions about discussing autism. We don't know whether it's autism or autisms. We don't know anything about the life history of the disease. There's discussions that 20% of kids lose their diagnosis. Is that the natural life history of the disease, or is that response to treatment? We know nothing about the benefits of treating the GI disorders, of treating the redox abnormalities, whether there's unifying metabolic disorders. The question of whether there's toxicity of vaccine components, not in the general population, but in vulnerable individuals. We don't even have good data on the prevalence of regressive versus early onset autisms. And without that kind of information, it's terribly hard to have a discussion. So I'm going to share with you some of the questions that I'm trying to understand in our work. And I'm going to proceed from the general to the specific. So first, whenever we think about a disease process, we're thinking about some kind of an avalanche. There's a boulder at the top of a hill. It starts to roll down. It gets more rocks. Eventually you destroy the village. Well, the measures that you're going to take depend upon where you are in the avalanche. If you intervene early, all you have to do is stabilize the boulder. If you have to rebuild the village, you have a very different problem. We have no idea where we are when we see a child with autism, where we are in the avalanche. We don't know whether there's multiple avalanches or stages occurring at the same time. We need to think also about resilience theory. Resilience theory has been best studied in ecology, where people have worked out a lot of the mathematical ideas looking at how you convert a clear lake to a lake that's overgrown by algae. And the way that this happens is that you have phosphate concentrations increasing, and they get to a certain point, and the lake flips into an algal overgrowth situation. For a long time, this was thought to be a chaotic shift. We now know this is a property of complex systems. Any system that obeys the mathematics of complex systems, which we all do, which all biological systems do, will behave like this. Well, the system is resilient against stressors, but when you get into the other state, that's resilient on the way down, too. And that's why it's so hard to clear lakes that have become overgrown with algae. You can't get the phosphate concentrations low enough. Well, is regressive autism a case of resilience failed? And if so, what are the implications for treatment? What are the equivalents of getting those phosphate concentrations so low so that we can get back to some more normalized status? We don't know enough about the problems of big changes versus cumulative small changes. Development is a cumulative interest problem. And to illustrate to you how important small changes can be, the upper line in this graph is what happens to progenitor cells in a situation where at every generation, 55% of them self-renew. And the bottom graph is what happens if 55% of them differentiate. In one situation, you increase progenitors, in the other, they go to extinction over simply 15 cell cycles. And when you look in vivo, you can't detect differences like this. You can't detect these 10% differences, but you get this big readout at the end. We don't know enough about that. Now, we do know that all disease is based on cellular dysfunction. And although it's very interesting and important to have genes that we can work on, those genes mean nothing except in their cellular context. That is what gives them a role in biology. But what cells should we work on? We have a lot of cells in the developing nervous system that we now know about. That, of course, many laboratories work on stem cells, which are the ones on the top of this list, neuroepithelial stem cells, and many labs work on the cells at the bottom of the list, which are the neurons and the astrocytes, the myelin-forming oligodendrocytes. Sadly, there's only a small number of us in the community who work on the lineage-restricted progenitors that are in the middle, even though these are the cells that are the workhorses of building tissue. Right? The neuroepithelial stem cells, or any stem cell, are a tiny contaminant in the population. They generate progenitor cells which do all the division, the key decision-making about when I should differentiate, what I'm going to become, and studies on those cells, attempts to purify those cells, have been rather limited. We know of some of them. And we know through our studies of them, through work that my colleague Margot Meyer-Purcell and I have been doing for the past decade, studying things like thyroid hormone deficiency and iron deficiency, that many developmental maladies are diseases of precursor cell function. That they can come in different ways, generation of specific cell populations of differentiated cell types, generating enough numbers. And this is coming up whether we study environmental influences or nutritional influences or genetic influences. So we have to wonder whether autism may be caused, at least in part, by disruption of normal stem and progenitor cell function. I want to hasten to say, particularly because of the podcast arrangements, that this does not mean that autism is amenable to stem cell therapies. That one of the messages that I deliver when I speak at DAN meetings or other parent meetings is that the stem cell vultures, just as they have been circling and landing on those needy about spinal cord injury, those needy in cancer, those needy in cerebral palsy, are also circling the autism community. And there are many offshore clinics that for many thousands of dollars will provide stem cell therapies to treat autism. None of it has a scientific basis. And it's important that that word be spread. It may someday teach us something, but not yet. Okay, now I want to switch to the biology not of precursor cells, but of the astrocytes. Numerous studies have now indicated that there is a hypertrophy, a gliosis of astrocytes in the brains of autistic children. What does that actually mean in terms of thinking about the biology of autism? Well, to get there, I'm going to have to just tell you very briefly about some of our work on spinal cord injury. We've been trying to figure out how to repair the damaged spinal cord in collaborations with Stephen and Jeanette Davies at the University of Colorado. Like many people, we've been transplanting all sorts of things into lesions over the years. And what happens in most transplants is that the axons will grow to the edge of the lesion, this yellow area that's transplanted with cells. Most of them will stop and turn around. A small number will enter the lesion, and a smaller number will grow through. And we thought we could do better initially by transplanting the earliest embryonic precursors of the developing spinal cord. These cells make all the glial cells of the spinal cord. They ought to be great at this, but they're not. They don't do anything. What makes a big difference is taking these early precursors and turning them into a very specific kind of astrocyte. And when we transplant these cells, now 70% of the injured dorsal column axons enter the lesion site, and within 8 days, 70% of those exit the lesion site. Previously, the highest level of regenerative growth through a lesion anyone had ever seen was 5% of axons taking 6 months to get there. That's with the right astrocyte. So now we have to ask, what's the biology here? I'll skip these slides. So we now ask the question of what happens if we take this same precursor cell and make astrocytes in two different ways. The astrocytes we made before are made with bone morphogenetic protein, but we can also make astrocytes by exposing precursors to other molecules, members of the interleukin-6 family that work through the GP130 receptors. And if we make those astrocytes and transplant those, we get no regeneration. None whatsoever, and even worse, we generate in the experimental animals neuropathic pain syndromes, syndromes of mechanical allodynia and thermal hypersensitivity, the kind of syndromes that, of course, in the field of spinal cord injury repair would be devastating. Now, we have a deep concern now that the kinds of cytokines that are involved in generating these bad astrocytes are exactly the cytokines that are elevated in the cerebral spinal fluid of children with autism. So are the kinds of gliosis that are being generated in these kids themselves inhibitory of the kind of repair regeneration that we would like? What are the implications of this question? What are the implications in terms of trying to control inflammation and what benefit we might get from that? Okay. We also know that many children with autism are more oxidized than normal. Is this important in pathogenesis? So continuing on from what Beth Ann told you, we're very concerned about electron transfer, bioenergetics, and critically with the fact that not all oxidation is oxidative stress. It's a critical component of normal regulation of how cells function. We've known for a long time that antioxidants will block cell death. We got interested in this because we were trying to figure out how to prevent myelin-forming oligodendrocytes from being killed by tumor necrosis factor, another cytokine that's elevated in the cerebral spinal fluid of autistic children. And for various reasons, I hypothesized that a drug called N-acetylcysteine, which is a cysteine prodrug, it's readily taken up by cells, it's converted to glutathione. Glutathione is your major redox regulator. We predicted that this would block killing, and that is indeed what we found. So here in the lower right-hand graph, the white bar is killing by tumor necrosis factors, and the colored bars are various levels of now protection. Okay, so it's a heavily cited paper because it was one of the early discoveries that you could use antioxidants to block cell death. But the key part of the paper, which people have ignored, is this. And this is what has me so interested in terms of autism and other areas of oxidative stress. The ability of a cell to respond to its environment is regulated by that cell's redox state. So here's an example where we're taking spinal ganglion neurons and we're exposing them to nerve growth factor, the best studied survival factor for these cells. The purple bars show you nerve growth factor alone. The green bars are plus N-acetylcysteine. What you can see on the lower end of the dose spectrum is that if you don't have very much nerve growth factor, N-acetylcysteine doesn't do anything, and N-acetylcysteine by itself doesn't do anything. But if you go to the other end of the spectrum, you get, by adding N-acetylcysteine, a 1,300% increase in the number of surviving neurons, but only a 15% change in redox state, a 15% increase in glutathione. So 15% increase in glutathione, 1,300% benefit in terms of cell survival. Now, anytime you see a system, be it biological or financial, where you put in $1 and you get $80 back, you want to know more about it. I have not been as successful in investigating the financial systems as we have been with the biological ones. What we've learned over the years is that redox state controls precursor cell function. Classical signaling molecules that enhance self-renewal or induce differentiation converge on changing redox state as a necessary part of their mechanism. And the organism even uses developmental regulation of redox state to control precursor cell function, the timing of development in different regions of the nervous system. And there's rules that, at least for the oligodendrocyte lineage, are quite clear, that cells that are more reduced are more responsive to mitogens and survival factors. Cells that are more oxidized are more responsive to differentiation and death factors. For those of us like myself who are interested in tissue repair, of course, this is of great concern because tissue repair is an oxidative stress, tissue injury is an oxidative stress situation. So in order to get repair, do we have to do a metabolic reprogramming to make the cells more reduced? But the question for autism, I think, is whether this being more oxidized creates a vulnerability phenotype. Does that mean that now these cells are more vulnerable to toxins in the environment, for example? Well, that's not the only area in which Radix State contributes to changes in cell functions. There's some very excellent papers in the literature on Radix State and its control of the immune response. One example is that oxidized macrophages promote a Th2 response. We know from the literature that children with autism spectrum disorders tend to be in a Th2 imbalance. Could that be just because of being more oxidized? There's some good data in the literature that suggests that that is so. And sadly, being in a Th2 lock-in would lead to production of some of the cytokines that we're very worried about. Okay, well that brings us to toxicology. And we got interested in toxicology for a large number of reasons. Part of it was this wonderful challenge that you've heard spoken of all of these chemicals that we know nothing about. So we don't know what to do about them. What a fantastic challenge to work on, particularly with the World Health Organization now estimating that 30 to 40% of the burden of childhood disease is due to environmental factors. Some of it undernutrition, some of it toxicant exposure. So how do we study this? Well, we know that many environmental toxicants are potent pro-oxidants. So we go to the literature, particularly to the work of people like Mickey Ashner here. We say, what have people learned about different toxicants? And we started our work with methylmercury. And the work of Mickey and others had shown that astrocytes, shown here, have an LD50 in the range of about 500 nanomolar when you give them methylmercury. So that's how we started. And then we went to the progenitor cells that we work with, started looking at their sensitivities, and what we find is that oligodendrocyte progenitors, embryonic glial restricted progenitor cells, oligodendrocytes also have LD50s that are very, very much lower level. So the LD50 for oligodendrocyte progenitor cells is about 25 nanomolar. That's 5 parts per billion mercury. That's the level of mercury you have in your bloodstream if you eat tuna fish 3 times a week. And the human brain concentrates mercury 5 to 7-fold over the bloodstream. So studying progenitor cells, we get a very, very different view of what is dangerous. Now, but we don't know much about those areas, so first we have to ask whether our initial starting hypothesis was even right, that we should be worried about oxidative changes, and yes, we are. 20 nanomolar methylmercury is enough to oxidize oligodendrocyte progenitor cells in exactly the range that we're interested in. So then we can make predictions that we predict that you're going to knock progenitor cells out of division. So these are 3-dimensional representations of clonal growth. And if you go along the back wall, what you're looking at is the number of progenitors in a clone. The left wall is the number of clones with any given, is the number of oligodendrocytes in a clone. And the height of any bar is the number of clones with any given composition. So the upper bar, there's lots of division. The lower bar, there's less division and more differentiation. All we've done is expose cultures to 20 nanomolar methylmercury. So that's as we predict. We predict that being more oxidized makes you more vulnerable to other stressors. So here's an experiment where we take oligodendrocytes, we expose them to tumor necrosis factor in the blue bars, dose that doesn't kill very much. We expose them to methylmercury, and over here at this end of the graph, if you can see that, do you see the cursor? Yes. Okay. Where there's no tumor necrosis factor, the methylmercury doesn't do much, but add them together, and now you get this significant killing. And we are seeing these synergies with every insult we look at. We look at oxygen glucose deprivation, we look at glutamate, we look at chemotherapeutic agents, and the ability of environmental toxicants to increase sensitivity to these other insults is enormous. So how does this work? Well, I suggested to one of my postdoctoral fellows, Dr. Zybo Lee, to take on the challenge of dissecting platelet-derived growth factor signaling from the nucleus back to the receptor in cells exposed to methylmercury. And that this work is published. I'm just going to show you the summary. So the first experiments that he did showed here with the red boxes that when he looked at response to platelet-derived growth factor in cells treated with methylmercury, that everything that we looked at was suppressed. Transcription in the nucleus from the serum response element and NF-kappa B promoters, phosphorylation of AKT and ERK as intermediate enzymes in signal processing, even phosphorylation of the platelet-derived growth factor receptor. So that was just as my signaling colleagues had told me we would find, that redox affects everything, it's global, you can't study it, give it up, go back to the things that you're good at, leave the signaling to us guys. And fortunately, we didn't listen. And we went on, and very fortunately, Zybo did the right control, which was to expose cells to neurotrophin-3. And what the white boxes indicate is that in cells exposed to neurotrophin-3, there was no effect of methylmercury or any other toxicant that we studied. So that suggested things were going on at the receptor level. So Zybo then looked at total receptor levels and found that specifically the levels of receptor for platelet-derived growth factor fell, and the levels of the TRAC-C receptor for neurotrophin-3 were unchanged. And that was the point at which Dr. Lee exercised one of the most important scientific principles that you young scientists here need to learn, which is that sometimes 8 or 10 or 12 months of hard work in the laboratory can save you as much as 45 minutes in the library. Fortunately, Dr. Lee uses the library, and we very quickly got to this discovery, which is that small increases in oxidative status activate an enzyme called FIN. FIN is a member of the SARC family of kinases. It lives in the membrane. It has multiple targets. One of those targets is another enzyme called Sybil. Sybil's a ubiquitin ligase. It attaches monoubiquitin to proteins and targets them for degradation. And some of its targets are receptor tyrosine kinases, very specific receptor tyrosine kinases. So when you oxidize a cell, and it doesn't matter how, whether it's genetic, whether it's pesticides, whether it's organic mercurials, whether it's chemotherapeutic agents, whether it's amyloid beta protein, you turn on this pathway, you degrade these receptors, and one of the targets of Sybil, coincidentally enough, so interestingly, is c-Met. So here's another pathway to get you to the same endpoint that Pat talked about before. Lower levels of c-Met, but now not associated with a mutation in the promoter, associated with being more oxidized, and with falls on these other receptors also. And I should say that everything that I've been telling you about, we do this both in vitro and we do it in vivo. And the in vivo results are as we predict from what we see in a tissue culture dish. Okay. Now, we've also looked at thimerosal. And I want to make the point that thimerosal is not special in either direction. Biologically, it is exactly as toxic as methylmercury in our hands. We see no difference, which means that at these very low exposure levels, we see these changes. I can take precisely the papers that Dr. Ashner presented, and I can argue what I think is a convincing case to you, that what this data shows you is that thimerosal levels in the brain are exactly in the range that we are worried about, and that those studies that have been claimed to support the safety of thimerosal have no value whatsoever and can be readily interpreted in support of the hypothesis that when thimerosal came out of vaccines, the biological prevalence of autism dropped. This is a very serious problem. I want to be clear what I'm saying. I'm not saying that I think that thimerosal causes autism. I'm saying that the data that we use to take positions on this is not good. And we need to really pay attention to this in some of the ways that have been alluded to by our epidemiological colleagues. I also want to point out on this graph something about vulnerability that has not come up yet. So we've discovered that different strains of mice, that cells isolated from different strains of mice, have different levels of vulnerability to mercury and thimerosal and all sorts of other things that we're studying. And what you see on this graph is that the upper line is oligodendrocyte progenitors isolated from CBA mice. The bottom line is oligodendrocyte progenitors isolated from SJL mice. And not only do the SJL mice get injured at lower exposure levels, the transition from a little injury to a big injury occurs much more quickly. So it's not simply that you're more vulnerable to lower levels. The dose-response curve looks like it gets truncated. And whether this is a part of understanding autism, we don't know yet. So now in my last couple of slides. At the level of individuals, what we are particularly interested in is understanding whether metabolic regulation is critical. The work that we're doing with Jill James in particular. Is overcoming gliosis critical? We desperately want to understand this vulnerability phenotype, a interest that we share with Isaac, as you've heard. We want to understand the avalanche, of course. But there's also other things that we are wondering about. And there's one that I want to raise, which is in the area of early intervention. One of the most robust findings in children with autism is myelination phenotypes. And the myelination disorders in these kids are quite intriguing. They've been well characterized by Martha Herbert and her colleagues, but there's also studies out there on the auditory brainstem response, which measures the speed of impulse conduction from the ear back to the brain. And the abnormalities that the kids show are consistent with a general failure of myelination, of generation of sufficient myelin. Now, the kinds of changes that you see when that happens theoretically can disrupt phoneme processing. And if you disrupt phoneme processing, you have real troubles with language acquisition. So an interesting experiment I've been talking about with people is if we had kids who were recognized early as being at risk or starting to show these problems, and we were to take them, and instead of insisting that they learn auditory language, we start immediately intervening to teach them sign language, would we have a much better opportunity for giving them language acquisition at that time of cortical development when it is critical that the human brain acquires some language, any language, it doesn't matter which. Now, we don't know the answer to that, but we know that the visual system does not do phoneme processing the way that the auditory system does. And as I go around and I give talks at various places, I have therapists coming up to me and telling me that they notice that when they try and communicate with kids with sign language, that for some kids it actually works much better. Easy experiment to do. And last, at the level of society and the scientists, and this is the same slide when I talk at DAN meetings. Data, data, data. Chelation is used widely in this community. As you've heard, and I think Mickey politely understated, there's no data out there of any sort. If you want to argue that chelation is bad or good or worthless, the data is just not there. The epidemiology is a problem. We don't understand what all the changes in diagnosis mean for the changes in prevalence of autism. These ideas that these studies from Denmark, for example, show an increase in prevalence of autism, it's just not, to my mind, it's just not true because the diagnostic standards changed, let alone the fact that in 1995 they added 6 times as many patients to their analysis but kept their denominator exactly the same. Right? This is not good scientific procedure. And we need to educate our colleagues in the government as to what the costs of this are. This, however we got here, whether this is changes in diagnosis or whether this is a true biological increase, this is one of the most prevalent childhood disorders. It is more prevalent than childhood cancer. It is more prevalent than type 1 diabetes. And we are spending far more research funds in these other areas than we are for these kids, many of whom are going to need lifetime care, where families need urgent help and we have the responsibility to help them.